Hello learners, so far you have seen a lot about biology, a lot about mathematics, but today in this chapter we are looking to another aspect of biology, that is mathematical modeling of mutations. Ever wondered how tiny changes in our gene can lead to such big differences? And today we are going to see all these changes. Now picture this. Mutations are like nature's tiny experiments. They are the reasons why each of us is unique. In this part of our journey, we will discover why understanding mutation matters. From adapting to a new environment to dealing with diseases, mutations hold the important key. Now, before I go ahead, I will describe the brief overview of mathematical modeling in genetics. Now imagine, you have a puzzle but some pieces are missing. Mathematical models are like those missing pieces. They help to complete the picture. In genetics, mathematics helps us predict how mutations spread, why they happen, and what are their effects. In this part, we will take a quick tour of how math and genetics come together and perform a powerful duo to understand all these complicated methods. Now, I will very briefly describe the types of mutations. First, all of you must have studied in your UG program about point mutation. Now, let me explain what are point mutations. Imagine altering just one letter in a book and suddenly the whole story changes. Point mutations are like that. They involve swapping, adding, or removing a single genetic letter. Now, join us as we decode the impact of these small changes and see how maths helps us keep senses of their effects. Another types are insertion and deletion. Now, you ever tried to solve a jigsaw puzzle where some extra pieces are missing? Now, that's insertion and deletion to our genetic codes. These mutations either add a small section or remove various sections from these genetic codes. It is like editing a recipe by adding or skipping a lot of ingredients. Another important type is chromosomal rearrangements. Imagine shuffling the chapters of a book. The story would take a new twist. Chromosomal rearrangement do something similar to our genes. They shuffle large chunk of genetic material between different chromosomes. Now you have understand about these editing methods. I will explain very briefly about molecular basis of mutations. First, how these mutations arrive at our system. Now you imagine you have seen a YouTube video. From there you have copied a recipe. Now you are making cake in your home. What happens is exactly how you have seen in those videos you try to replicate in your kitchen. But imagine sometimes the quantities of these ingredients might differ when you copy in your kitchen. Now what happened is your recipe won't exactly look or won't exactly taste like what you have anticipated from that video. The mutations are exactly like that. Whenever organisms multiply or they duplicate each other, they often copy their genetic codes and replicate to a newer system. In this method, there is a possibility of errors while copying these DNA segments. These errors finally leads to what we call today as mutations. Now, there are other factors like environmental factors. They also influence mutation. Our genes are like dedicated artwork influenced by environment around us. Imagine a painting fading in sunlight. The environmental factors also affects our gene. From radiation to chemicals, these factors can trigger mutations. Okay, so far we have learned what are different kinds of mutation and how they arrive. One very important factor is mutation rate. I will briefly describe about these mutation rates. Mutation rates are like speedometer of genetic changes. Mutation rate tells us how quickly these little genetics or these changes in our genotype occurs. Imagine a typewriter typing out a story, but sometimes it slips up and hits a wrong key. Mutations are like rhythms, and the speed of typing represents the mutation rate. Factors like mutation rate really the factors affecting mutation rates. Now I will briefly explain this part. Just like real explorers, they face challenges along their way. Environmental factors, chemicals, and even the way our DNA gets copied can speed up or slow down these mutation rates. It's like genes taking a different path to their adventure. Now, we will slowly dive into the mathematical modeling part. Now, imagine you are a wizard creating a virtual world on your computer where you can predict everything from how your ice cream melts to how your plant grows. The mathematical modeling is exactly like your wand, and in this exact the entire picture you can imagine as mathematical modeling of predicting things even before they occur. Let me give you a very funny example of how this model works. Suppose you are hosting a party. In that party, you know how many guests are coming. Now, in among these guests, some of these guests will be kids, some of these guests will be adults, and you know exactly by an average how many cookies will be consumed by these kids and how many cookies will be consumed by the adults. So you will have a generic idea of average number among the different age groups. Now you can not exactly but approximately predict how many cookies you need to bake to host that particular party. The mathematical modelings work in a similar way. You work with different equations, you work with different principles. Based upon that, these equations are being created, and based upon all these segments, you will predict the effect of any genetic changes. In this case, any mutation, what it can occur, what will be the output of these small changes, you can exactly predict that. One common mathematical model used to capture the accumulation of point mutation in asexual or bacterial population is Kimura's two-parameter model. I will briefly explain this model. This model describes the evolution of DNA sequences over time. Consider both transitions, either changing from purine to pyrimidine, it's A to G, or C to T, and the transversion between the purines and pyrimidines as well. The equation exactly depicts as it is shown in the picture, just like delta P equals to mu times P times 1 minus P minus 2 times mu q P. Now each part, I will explain the importance of each part. Here in this equation, delta P is change in the allele frequency, mu is the mutation rate that we have learned just now. P is the frequency of the original allele and Q is the frequency of new allele. By this, you can exactly predict the status of these alleles, or in this case delta P. I will also briefly explain about the Hardy-Weinberg equilibrium. Now, it's like a genetic harmony that keeps the population in balance. Imagine a colorful garden filled with different flowers. Now, if no flowers grow and no flowers disappear, the garden's beauty remains constant. That's the essence of Hardy-Weinberg equilibrium. It's a snapshot of genetic world where genes peacefully coexist over time. It's like a theoretical representation where no mutation happens at all. Now, I will briefly explain the mathematical representation and its effects. Think of it as a secret code that reveals the genetics of that flower. This code is a mathematical formula, a bit like a decoder ring that helps us understand the frequencies of different genotypes in the group of organism. Here is the exciting part. Hardy-Weinberg equilibrium is like a genetic compass. When gene frequencies matches the prediction, it's like a compass needle pointing a true north. Population The population's genes are in synchronous condition, but if the gene frequencies start to dance around in different direction, the compass needle wobbles, signaling changes are happening. Imagine a group of animals in an Iceland. Each of these, they will have their own stack of food. If suddenly some animals start munching on a new treat, it's a sign that equilibrium is disrupted. The mathematical representation of is p square plus 2pq plus q square equals to 1. I will briefly explain what are the different components. The p represents the frequency of one allele. It's usually the dominant allele what we are talking about. The q represents the frequency of the other allele. Usually it's the recessive allele. So the p square and the q square represent the allelic frequency of homozygous condition in the dominant or in the recessive allele, where 2pq is the frequency of heterozygous individual. This equation reflects the stable gene proportions in a population under certain conditions. Imagine no mutation, no migration, no selection, or no genetic drift is taking place. This equation shows the ideal condition where the Hardy-Weinberg equilibrium is being maintained. It's a fundamental concept in population genetics, and it helps us understand how genetic traits are passed from one generation to the next generation in a stable population. Now, we have learned so far about different types of mutation and different mathematical aspect of it. I will briefly explain how we can model a point mutation. Now, we have learned that single nucleotide changes are denoted as point mutations. These tiny changes can rewrite the entire genetic story. Imagine you are editing a sentence in a book. Changing just one word, that's what a mutation does. It swaps a single genetic letter. We are about to unravel the magic behind these letters, that swaps, and how math can help us predict their effect. So mathematical representation is the p of ij function of time is equal to half plus half of e to the power minus 2 times mu t. This equation helps us predict how likely a genetic letter that will change letter from I to J over time. The symbols seems may like magical runs, but together they unveil the hidden language of genetic mutations. Genetic mutations are very complex to understand. Some of these effects are beneficial for us, some of these effects are highly deleterious for us. So in a brief way, let me explain about beneficial mutations. So these are the bright side of these mutations, the beneficial ones. Imagine finding a secret shortcut on your way to school. That's what beneficial mutations are like. They are the genetic changes that brings the positive advantage. It's like acquiring new skills that helps you to perform any function in a better way. Similarly, the opposite side of it is deleterious mutations. So imagine a typo in a recipe that turns your cookies into bricks. So this is a funny example, but that's exactly how these deleterious mutations are like. Deleterious mutations are like genetic blunders that brings negative effect. They can lead to a healthy individual to a diseased one. They are like the villains in the movie. They often brings a lot of disadvantages in a healthy individuals. Beneficial mutations are often modeled as using the equations that describe how the frequency of the beneficial mutations alleles changes over time due to an advantage it provides. A simple model look like this, as it is explained in the equation. Delta p equals to s times p times 1 minus p, where delta p is a change in the frequency of beneficial allele, s is a selection coefficient that represent the advantage of beneficial alleles over the other alleles. P is the frequency of beneficial alleles. This equation suggests that changes in frequency of the beneficial allele is in proportion to both the selection coefficient and the existing frequency p. Similarly, let us see how the deleterious mutation can be modeled. So, of these deleterious mutations often leads to reduction in the fitness. That's why the equation here explains the negative side of it. So, the equation is delta p equals to minus h times p times 1 minus p. As you can see, it is exactly just like a beneficial mutation where your fitness is increased, where in this case the fitness is decreased. Again, the delta p is a change in the frequency of deleterious mutation. H is a dominance coefficient indicating the extent of which the deleterious mutation is expressed in a heterozygous condition. P is the frequency of deleterious allele. This equation suggests that changes in the frequency of deleterious allele is proportional to both the dominance coefficient H and the existing frequency P. There are a lot of work has been done to model these beneficial mutations as well as these deleterious mutations. Now, the bright side of it, how we are different from each other and how we have evolved over the time. It is due to the accumulation of these beneficial mutations. So, in a laboratory setup where you can work with microbes or yeast, you can grow them in a specific condition. Now, these specific conditions provide different S, that is selection coefficient or the selection pressure. Selection pressure will decide how the uh, how the evolution of these acquired characteristics takes place. Now, in a laboratory setup, you can exactly see after generations of evolving these bacteria or yeast how a specific trait has been gained. There are a lot of experiments that is being performed across the world. You can see that modeling beneficial mutations is not a problem, but modeling deleterious mutation is a real challenge. These deleterious mutations often represent a reduction in their fitness, and because of that you will no longer have those kind of organism in your system. Okay, so it's easy to see and model a beneficial mutation rather than a deleterious mutation. Now, I will briefly explain about mutation accumulation models. First, let me tell about hitchhiking. What is a hitchhiking? You might have heard a word about hiking. Suppose a traveler wants to travel from one location to another location. In this way, he wants to limit his resources or consumption of money. So what he does is he takes lift from different people, and in this way he travels from one location to another location. Here he get down and ask another person to give him a lift. In this way he will reach his destination with the help of different travelers. This is exactly how in our genetics the mutations also travel. Suppose in a bacterial case or in a yeast case, you are exposed to an environment where selection pressure is working. In that case, you have acquired a lot of good mutations, that means beneficial mutation, and in the same pathway simultaneously you have also acquired some of the deleterious mutations. But but the the effect of beneficial mutation is so large that the effect of deleterious mutation is masked. So during this adaptation or during this evolution process, the deleterious mutation is a kind of lifted from from one generation to another generation. This is exactly what happened in hitchhiking. Another important parameter is called as Mueller's ratchet. Just like a ratchet which only travels in a unidirectional flow, it is exactly like that. So consider a population where a fixed size of organism, where each generation mutation occurs, and each mutation introduced a small deleterious effects. Over the time, this population have accumulated enough number of deleterious mutation because there is no genetic recombination to remove these mutations. It's like a simplified mathematical expression that captures the essence of Mueller's ratchet. Here in these equations, we can exactly model the Mueller's ratchet. It's delta n equals to n times 1 minus e to the power mu s, where delta n represent the changes in the number of individual in that particular population. N is the effective population size, mu is a mutation rate per individual per generation, and s is the selection coefficient representing the fitness reduction caused by each mutation. E is a natural logarithmic function. This equation suggests that changes in a population size is proportional to the population size itself and the accumulation impact of deleterious mutations. As deleterious mutation accumulates generation by generation, the population size can decrease over time. In a lot of diseases, it is being observed that they are effect of accumulation of these deleterious mutation. This is the wrong side, but the bright side is the entire evolutionary process is based upon the accumulation of these beneficial mutation. I will give you a very brief example. I have one number of individual who can make a long jump for about 4 feet. Now imagine there is a slight mutation takes place in that particular organism, and it can jump higher than 4 feet, like 5 feet, 6 feet, 10 feet, something like that. So it's not just one generational work. So the beneficial mutation which are making these organisms to jump in an efficient way are accumulated over the time, and you can see from these accumulations now the organism is able to perform in almost every environmental condition. I will also discuss a brief example where you can imagine our stomach. So our stomach is having a digestion system. Now imagine if your stomach system is only designed to digest a particular food, say for example rice, then what happened to the different other kind of foods? So over the time where these organisms start utilizing different food habits, there are genetic changes that occurred inside that particular organism, and that helps them to digest those different kinds of food. This is same thing happening in case of accumulation of either of beneficial mutation or the deleterious mutation. I will also explain the brief of I will also explain briefly about the evolutionary consequences of this mutation. Now, role of mutation in adaptation and evolution. We will have a different dedicated chapter regarding this particular topic, but since it is the mutation is concerned, it will be incomplete without talking about the adaptation and evolution. Imagine a wardrobe full of different outfits. The mutations are like adding new clothes to keep up the changing trends. They play a very vital role in adaptation and evolution, allowing species to adjust to their surrounding over time. Think of it as nature's way of giving creatures the right tool to survive. So, we have explored a lot of different phenomena about that mutation, how they work, how to model them, what are the different effects. In the same process, I will also explain about the punctuated equilibrium versus the gradualism. Now let's talk about the speed of these changes. So imagine a evolution as a story. So punctuated equilibriums are like moment of intense, the drama, or that followed a period of calm. So it's like a roller coaster with sudden drops and slopes that is followed by slow climbs. So on the other hand, the gradualism is like a steady sterile where the changes happen slowly over the time. It's like a painting a master stroke step by step. Okay, so learners, we have learned a lot of things about these theoretical concepts that exist Now, in the science, unless and until you validate these theoretical concepts with experimental tools, so unless we justify these theoretical tools with the help of experimental setups, you cannot say very confidently about these theoretical concepts. I will explain about these experimental validation parts. So in our previous lectures we have learned about different animal models, so or different bacterial models. So they are the perfect tools to validate these mutational models. For example, you take example of E. coli. E. coli generation time is 20 to 30 minutes. So evolution is a very slow process, and occurrence of these mutations are like ladders for these evolutionary processes. So imagine you are baking a cake from a new recipe. Now what you want to do is you would like to taste it and make sure it's just right. Similarly, we validate mutational models with real experiments to ensure if they are accurate. In genetics, we might use bacteria in a lab to mimic the mutation and see if our model prediction matches the real outcomes. Okay, so just now I talked about bacterial growth. Bacteria are the perfect agents for these kind of experiments because of their, I just mentioned, their uh, low duplication time. For example, take example of E. coli. E. coli takes 20 to 30 minutes to duplicate its entire genetic content. So whenever we have introduced a mutation or naturally it has occurred to that particular individual, we can track that what happens to that particular mutations. In the current scenario, there are tools which tags these genetic changes with fluorescent dye, or there is another method called as DNA barcoding. They exist, you can track that particular organism. So let me talk about this. This is one individual that is having a particular beneficial mutations in his DNA. Now it start multiplying. When it start multiplying, the entire population will be replaced by this individual which is having a beneficial mutation. You can exactly track how these individuals are growing inside that particular environment. Suppose in the if I continue this process and another mutation occurs, and you have certain techniques by which you can track the another mutation. What you can do is you can exactly model how the new beneficial mutation increase the frequency of that particular organism and it start multiplying, and how quickly it can outgrow the previous individual. Similarly, how the previous individual's number is decreasing over the time, you can also predict that. So these are the perfect examples which shows the experimental validation of exactly your model prediction. So in a real life example, you and I, we are in continuous learning phase. So for example, we must have crossed our class 12, and from there onwards we have been studied the specialized subject, something like that. These organisms learn from their environment, what are the environmental condition and how perfectly they can grow in that particular environment. They learn from the environment and they incorporate a lot of changes. These changes are sometimes beneficial in nature, sometimes deleterious in nature. Let me tell you exactly figures. More than 95% of these mutations are either deleterious mutation or neutral mutation. Only 3 to 5% of these mutations are beneficial in nature. So you can see how difficult is to model these organisms and actually see inside the lab. So in this process, let me talk about the future directions in this particular area. Now, advancements in the mutation modeling have taught us a lot of different things. Now imagine the evolution of technology from a landline phones to smartphones. Mutation modeling is of similar journey of advancement. New tools and techniques are emerging, allowing us to model mutation with even greater accuracy. Think of us think of it as upgrading from basic recipe to high-tech kitchen full of gadgets. We will explore how these advancements are shaping the future of genetic predictions and understanding. Now, I will in today's lecture by slowly I will in today's lecture by telling you guys about the genomics and personalized medicines. Now, picture a tailor sewing a custom fit clothes. That's what integration with genomics and personalized medicine does for mutational models. As we, as we decode the genetic code of individuals, the mutation models can help to tailor these treatments and intervention based on the unique genetic information that is provided by us. Now, genetics and omics are not two very distant subjects. They often help each other to understand these concepts. In the next lecture, we will also learn about the different mathematical model of evolutionary processes. So far, we have learned about the mutations, and I have also told you about how these mutations makes a ladder to understand different evolutionary processes. Similarly, we will also look about the different diseases that occurs because of these point mutation. In we are living in the area of Chhattisgarh, and one very common disease, the common I would say blood disease, is thalassemia and the sickle cell anemia. You will be surprised to know that the sickle cell anemia is a result of just one point mutation in the entire billions of genetic codes. So you can see how important of these studies will be. We will be adding a lot of mathematical concepts and a lot of modeling concepts in our journey, so that you will be able to learn the biology, the essence of biology, essence of mathematics, and how computationally you can model all these real life problems. Thank you very much. Happy learning.